Welcome to CSEC Biology TCP. Today we're looking at the 2024 prep. We're looking at the January 2024 exam, and to be exact, we're looking at question number 5. You want to look at the other question we would have posted before, and we're going to be posting some after this presentation. But the paper was a treat if you were supposed to ask me. But this question was really interesting. I'm going to be looking at some interesting thing about this question, which more persons or most persons would be thinking that the question would have been on a biology paper. So for me, I like in the final prep to do my bio and HSB preparation together. Let's look at this question. Say the number of chromosomes in a human cell at the end of each of the following type of cell division. Now, mitosis pretty much results in the diploid number of chromosomes. That's a full number of chromosomes. So it would have 46 chromosomes, while meiosis result in the haploid, which is half a number. So it's going to be 23 for meiosis. Now, Bernadette is viewing two cells under a microscope. She concluded that one cell is in the metaphase stage and the other cell is in the telophase stage of mitosis. Describe what she would have observed in each cell that led her to decide, led her to have this conclusion. All right. When I teach this, you probably have never seen my PowerPoint meiosis kit, but I usually teach my students like this: I P mat C. So I interphase come before mitosis. Mitosis is actually prophase, metaphase, anaphase, telophase, and cytokinesis. Now, my way of teaching my kids to identify is to try to find metaphase first. M is for the middle, so you're going to be seeing the chromatids line up along the center of a spindle fiber. A, anaphase, you're going to see the chromatids moving to opposite end, they are moving away. And then telophase, that's for two, you're going to be seeing two new nuclear membranes being formed. That's the easiest way to teach my kids to remember the different phases or stages of mitosis. So here, metaphase, the nuclear membrane, of course, would disintegrate, and the chromatids, of course, would align themselves along the center of a spindle fiber. Then for telophase, we're going to have two nuclear membranes being formed around a set of chromosomes. And pretty much you'll see the cell getting ready for cytokinesis or for two new cells to be formed. Now, this is a rather interesting question. A lot of persons would be saying, oh, wow, this is an agriculture question. But remember, in reproduction, we look at both sexual and asexual methods of reproduction. And for the greater part, some teachers might not have gone into types of course, asexual propagation. If you have not and you need to know, you can visit my website and look for asexual reproduction, and you'll find a lot of methods to include parthenogenesis. All right, so Bingley enjoys the fruit from his friend Navindra mango tree. He asks Navindra for a seed so that he can grow the tree exactly like his. Navindra tells him that his plan will not work and suggests that he use another method to propagate a new mango plant. Now you're supposed to explain why Bingley plan will not work and suggest one alternative method by which he could obtain a mango tree exactly like Navindra's. Now, interesting to note, if he's going to be using the seed, that is a sexual method of reproduction. And what we know about sexual method of reproduction is that it leads to variation. So all the nice fancy stuff that he had in mind about that particular mango tree might not be obtained if a seed were to be used. And that's why we use the asexual method when we are reproducing things like ackee. Orange is really popular with this method, and of course mangoes. This would lead to variation in the new mango plant. Hence, the characteristic that he fell in love with might not be seen or tasted in the new plant that he would have grown from seed. Now, a good method to use is, of course, an asexual method of propagation, and commonly the ones used for mango would have been budding or grafting. So you have a whole lot of grafting, wedge, saggle, you name it, approach. But of course, you could look at budding or grafting. This brings us to another question that seems to be biology. Now, Karen has an orchard of trees of a same species. A disease attacked one of the trees and quickly spread to the others, destroying her orchard. Kyle has an adjacent orchard, but only a few of her trees get the disease and die. Using your understanding of genetics, you are supposed to explain why many of Kaylee's tree survived and why Karen's orchard was destroyed. This is not so bad. Now, the first thing we need to know is that Karen orchard was developed from asexual method of propagation, and that does not show variation naturally unless we have things like mutation. Hence, all the plant would be susceptible to the same disease as they were not able to adapt to the changes in the environment. Now, let us not be confused with the term adapt and natural selection. Organisms adapt to be able to survive with changes in the environment. On the other hand, Kyle or Kaylee's tree might have been propagated via sexual method using seeds. Now, this method have caused them to adapt and show variation. Hence, not all the plants would be affected by the disease, as those that are more tolerant would definitely not be affected by the disease. That's all we have for today. Until we next meet, please be reminded to like, share, and of course, subscribe. We do have some content on Amazon. If it's your wish to support us, you can go to Amazon and purchase one of our kids' book or one of our science lab manual. It will be linked in the ads that follow. Thanks much for watching, and please be reminded to study to show thyself